Hey guys, welcome to the 6th episode of Short Bits by Shorty. Just a heads up, Sunny might have a little sound clip somewhere in here, just to keep things interesting, if they aren't already. So watch out for Sunny. Second thing, also heads up, I'm not going to draw as many structures on these videos because there's too many, and I kind of want to just highlight the structures of the common scaffolds and their naming conventions, or any structures that were really emphasized in class. But if I'm wrong about not memorizing all the structures, feel free to slap me anytime in school. Yeah, let's get started. Okay, so just for kicks and giggles, and since I don't think he'll ask about this, but just so you have foundational knowledge of what a mycobacterial cell wall looks like, here it is. Outermost layer, you have the mycolic acids containing glycoproteins and porins. Under that, in going inside the cell, you have the arabinomannan layer. Blue, more inside, you have the peptidoglycan layer. And between that, you have lipoproteins. And then within that, more inwards in the cell, you have the cell membrane. And throughout the mycobacterial cell wall, you have a section called a lipoarabinomannan layer, which helps the fungi or mycobacteria evade immune responses, which includes them inactivating macrophages or scavenging oxidative radicals. So, since the kicks and giggles are done, let's go to the actual drugs. So first we're going to talk about isoniazid. So isoniazid's target is the mycolic acid part of the cell wall. And specifically, isoniazid targets mycolic acid biosynthesis, and this biosynthesis happens through type 2 fatty acid synthase, or FAST2s. And these FAST2s include INHA enzymes and enoyl ACP reductase enzymes. So the mechanism by which mycolic acid is produced in the mycobacteria cells is by a simple subunit. A simple subunit of mycolic acid is the beginning precursor of the mycolic acid cell wall, and this subunit is extended to the whole mycolic acid cell wall by INHA enzymes and enoyl ACP reductase enzymes to form longer mycolic acid chains. So the mycolic acid part of the mycobacteria cell wall is extended from a few subunits to a lot of subunits by INHA enzymes and enoyl ACP reductase enzymes. And this is further extended by the use of NAD plus and NADH. Remember that NAD plus has a plus in it, and NADH does not have a plus in it, if you want to remember that. So NAD plus is reduced to NADH, and the cycle of mycolic acid cell wall extension is continued. So isoniazid blocks the continuation of the extension of the mycolic acid chain or cell wall portion by binding or reacting with NAD+. But first, in order to react with NAD+, isoniazid is a prodrug that is change the radical form. So you can see a radical right here, which means an electron right here, by the CatG enzyme, which is a heme-containing peroxidase. So CatG is an enzyme, or specifically a peroxidase, that forms radicals by a heme portion or an iron-containing portion. Then that's what heme-containing peroxidase means. Then this radical reacts with NAD+, and therefore you get no mycolic acid growth because you stop the step of NAD plus going to NADH, and NADH reacting with the subunit, and the subunit cannot be extended by INHA, and enoyl ACP reductase. So I wrote it out for you. Number 1, isoniazid is activated by CatG, which is a heme-containing peroxidase, and this forms a radical. So isoniazid is a prodrug. This active metabolite reacts with NAD+, and by reacting with NAD+, you stop the process. So I drew a line right here, which means to stop the process of reduction to NADH. And if you can't reduce NADH, then you can't extend the mycolic acid portion of the cell wall. And if you can't do that, you don't get cell wall formation, and thus the mycobacteria die. So the way that mycobacteria have developed resistance against isoniazid is by 2 mechanisms. One, by producing less CatG. And if you remember earlier, CatG is important in converting isoniazid from the prodrug form to the radical form shown here. So if you produce less CatG, then you get less ability of isoniazid to react with NAD+, and less ability to interfere with mycolic acid synthesis. Number 2, mycobacteria have become more efficient at producing NADPH from NAD+, so isoniazid has less time to react with NAD+. And you know that if you interfere with isoniazid reacting with NAD+, then you interfere with isoniazid blocking the conversion to NADH, and isoniazid is less efficient at blocking mycolic acid cell wall synthesis. So ethambutol is our next drug, and its target is the inhibition of arabinosyl transferase, which is involved in the construction of lipoarabinomannan. I don't feel like scrolling up, but if you look in the handout I provided along with this video like I always do, you can see that in the cell wall picture, lipoarabinomannan is a part of the cell wall, and it's also involved in mediating or avoiding the immune response. So if you inhibit arabinosyl transferase, which constructs lipoarabinomannan, then you inhibit cell wall synthesis, and the mycobacteria can't survive. This also enhances the permeability of ethambutol and other drugs, so it's frequently used in combination therapy. Resistance to ethambutol can develop when mycobacteria overexpress the arabinosyl transferase gene. And when you overexpress this gene, then you produce more lipoarabinomannan, since arabinosyl transferase is involved in constructing lipoarabinomannan. So you overproduce lipoarabinomannan at a rate higher than ethambutol can react or inhibit arabinosyl transferase. So result is a net production of lipoarabinomannan, since ethambutol can't react fast enough to compete with this overexpression of the gene.
Leroy Jenkins.
Hi, Sonny. So our next drug is rifampin. Its target is RNA polymerase, and resistance to this drug can develop when RNA polymerase mutates, so that rifampin can no longer bind and inhibit mycobacteria from doing their thing. So our next antimycobacterial is pyrazinamide. It's a prodrug that's converted to pyrazinoic acid, which is the active drug, by PNCA. So you can remember the structure of pyrazinamide because it has, well, it's one of the few structures in this exam, but it has an amide group. So pyrazin amide has an amide group. Funny, right? And it's converted to pyrazinoic acid, which has an acid group, by PNCA. And mechanism of action, IDK, because it's unknown, but at pH 5 to 6, which are acidic pHs, it can accumulate more in the cells, so increased accumulation in the cells, which means it's increasingly active. resistance to pyrazinamide can be formed when PNCA mutations occur, and if you have PNCA mutations, then you can't convert the prodrug to the active drug to prevent mycobacteria activity. Xanthomycin is our last antimycobacterial in this lecture, and its target is MER-X. So by inhibiting MER-X, you inhibit the production of lipid 1, and if you inhibit lipid 1, then you inhibit the production of lipid 2, and which is a component of the peptidoglycan layer. And as you know from the picture of the mycobacteria cell wall I drew first, not just for kicks and giggles apparently, let's see, you inhibit the mycobacteria cell wall synthesis. So by inhibiting MER-X, you basically inhibit mycobacteria cell wall synthesis. This drug is specific for mycobacteria, so researchers have tested it against other cell lines like bacteria or other species, and xanthomycin was found to be either inactive or only very weakly active. So basically, xanthomycin inhibits MER-X, which makes lipid 1, which makes lipid 2, which makes the peptidoglycan layer. And if you stop the production of the peptidoglycan layer, then you stop mycobacterial cell wall synthesis. Xanthomycin is specific for mycobacteria, so may or may not work for other cell lines other than mycobacteria. So the next section after antimycobacterials are the antiparasitics. So the first class of drugs within antiparasitics are the antimalarial quinolines. So you can see that the quinolines have a common scaffold. I drew these because it might be an exam because there's just a common scaffold. So a 6-membered ring benzene with a 6-membered ring with a nitrogen. So if you look in here, quinolines all have the 2 6-membered rings, one without a nitrogen and one with a nitrogen. So here's your quinoline, here's your quinoline, so quinoline. So the red indicates your quinoline scaffold. And remember, your quinoline scaffold contains 2 6-membered rings, one without a nitrogen and one with a nitrogen. And the way that your quinolines work is the following. So Plasmodium is a parasite that digests hemoglobin. And when you digest hemoglobin, you release heme, which is basically a group that contains iron. So if you release heme, you have free heme, which is actually toxic to Plasmodium. But to make it less toxic, Plasmodium uses an enzyme called heme polymerase to convert free heme to hemosin, which is non-toxic and non-soluble. So what quinolines do is that they inhibit heme polymerase, and if you inhibit heme polymerase, you inhibit the conversion of free heme to hemosin. So more toxic free heme builds up in the Plasmodium, and the Plasmodium die. So our next drugs are pyrimethamine and sulfadoxine. Both inhibit folic acid metabolism, and if you inhibit folic acid metabolism, then you inhibit DNA synthesis. And this happens because folic acid is important in nucleotide production. So if you inhibit folic acid metabolism, then you inhibit nucleotide synthesis, and then you inhibit DNA synthesis. So your next 2 drugs are atovaquone and proguanil. So these drugs, like pyrimethamine and sulfadoxine, are given together to make them more effective. Atovaquone is an ubiquinone reductase inhibitor, which inhibits and by doing this, it inhibits cellular respiration and electron transport, which are important in parasite survival. Proguanil is a dihydrofolate reductase inhibitor, and structure is shown here because it's a prodrug, so that's kind of important. You can tell the guanyl group by the string of nitrogens. So guanol is basically a string of nitrogens. Easy way to remember this is proguanol is a pro-drug and it has a guanyl group. So proguanol is a pro-drug that has a guanyl group. Cool name, right? It's converted to the active drug cycloguanyl by CYP2C19. Ooh, pop quiz! What other drugs are metabolized by CYP2C19? Ba-da-ba-ba-da-ba. Your PPIs, so like omeprazole, pantoprazole. Yeah, cool. So your next class of drugs are your artemisinin, including artesunate. Their target is heme, and as you remember earlier, quinolines also target heme, and hemes are important with plasmodiums. So, artemisinin are unique because they form toxic radicals by interacting with a heme, and this is especially useful in plasmodium. So basically, artemisinin target heme, which forms toxic radicals in plasmodium, and that's it for that. So last drugs are your metronidazole and tinidazole. So metronidazole or tinidazole through redox or reduction oxidation chemistry or reactions in the cell produce reactive oxygen species. And these reactive oxygen species interact or react with various cellular components within your parasites, and this interferes with nucleotide activity or DNA synthesis. So metronidazole or tinidazole by redox chemistry produce reactive oxygen species, and these reactive oxygen species interfere with DNA synthesis by interfering with nucleotides in the cell. Ooh, also important thing, metronidazole and tinidazole act on anaerobes because anaerobes use this mechanism of conversion with redox chemistry. So, just to review everything in a nutshell, hopefully we're still under 20 minutes. It should be under 20 minutes. If not, I'm so sorry. Here's a mycobacterial cell wall for kicks and giggles. Each of these drugs act on different portions of the cell wall to inhibit cell wall synthesis and therefore interfere with mycobacterial survival. Isoniazid interferes with mycolic acid biosynthesis which includes the INHA enzyme and enoyl ACP reductase. Specifically, isoniazid is converted to the active form by the CatG enzyme, and this active form reacts with NAD+. Remember, NAD+ has a plus in it, and reacting with NAD+, if you stop the cycle of mycolic acid biosynthesis, and you stop mycolic acid chain elongation. Resistance to isoniazid can be conferred by less production of CatG or more efficient production of NADPH from NAD+. So an isoniazid has less time to react with NAD+. Ethambutol targets arabinoyl transferase, therefore inhibiting lipoarabinomannan, which is an important part of the mycobacterial cell wall. This also enhances the permeability of other drugs, so it's a lot of times used in combination therapy. Resistance can be developed by overexpression of the arabinosyl transferase gene. Rifampin targets RNA polymerase. Ooh, surprise, resistance can be conferred by mutation of RNA polymerase, so that rifampin can't no longer bind. Pyrazinamide is a prodrug that's converted to the active form pyrazinoic acid by PNCA. Remember, pyrazinamide has an amide group. Mechanism of action unknown, but at acidic pHs, it can accumulate more in the cell, so it can become more active. Resistance can be conferred to, surprise, PNCA mutations. Sancomycin targets MER-X, which therefore inhibiting peptidoglycan synthesis, therefore inhibiting mycobacterial cell wall synthesis. This drug is specific for mycobacteria. Now your antiparasitics. Your quinolines are a class of antiparasitic drugs, specifically antimalarial drugs. And remember, the quinolines have a quinoline group, which includes two 6-membered rings, one of which has a nitrogen and the other doesn't have a nitrogen. These target Plasmodium because these digest hemoglobin. And by digesting hemoglobin, you release heme, and Plasmodium convert the toxic heme to hemosin, which is non-toxic and non-soluble, by heme polymerase. So quinolines target heme polymerase, and by doing so, more toxic heme can build up in the cell and kill Plasmodium. Pyrimethamine and sulfadoxine are given together usually, and these both inhibit folic acid metabolism, therefore inhibiting DNA synthesis. Also given together are atovaquone and proguanil. Atovaquone is a ubiquinone reductase inhibitor, therefore inhibiting cellular respiration and electron transport. And proguanil is a dihydrofolate reductase inhibitor. Shown here, remember guanyls have the guanyl group. And this is converted from the prodrug to the active form cycloguanyl by CYP2C19. Artemisinin target your heme group, like as found in your plasmodiums, and these, by targeting the heme group, they form toxic radicals. Finally, you have metronidazole and tinidazole, which target anaerobes because anaerobes use redox chemistry. So metronidazole undergoes redox chemistry to form reactive oxygen species, and these interact with cellular components that assemble nucleotides. So by forming reactive oxygen species, you interfere with nucleotide production. So that's episode 6 in a nutshell. If you have any comments, concerns, feedback, I always take them at pollyvdotcom/CHRISTIANRUI629. Let's see. Yep. So that's it. See you for episode 7 if you're watching it.